As the modern age progresses, medical advancements continue to thrive due to human ingenuity and a drive for development. Just as bacteria and viruses adapt to our immune responses, humans must also adapt their treatments for these diseases. That is why, since scientists discovered that DNA changes cause cancer, they have sought a simple method to correct these changes through DNA manipulation. Despite several gene editing methods developed over the years, none were quick, easy, and affordable until 2013. That year, researchers demonstrated that the gene editing tool called CRISPR could precisely and easily alter human cell DNA, revolutionizing cancer research and treatment. This discovery and development of the CRISPR-Cas microbial adaptive immune system into a genome editing tool represent the collective efforts of many scientists worldwide. Now, a timeline from the Broad Institute stated that the history of CRISPR began between 1993 and 2005 with Francisco Mojica at the University of Alicante, Spain. In 1993, Mojica identified CRISPR loci and recognized that these sequences shared common features now known as CRISPR sequences. In 2005, he hypothesized that CRISPR functions as an adaptive immune system in bacteria, a theory confirmed by finding matches between CRISPR sequences and bacteriophage DNA. Following in May 2005, Alexander Bolton discovered a novel CRISPR locus in Streptococcus thermophilus, which included a new protein, Cas9, with nuclease activity and identified the polyspacer adjacent motif, or PAM, a crucial for target recognition. In March 2006, Eugene Kuhn and NIH proposed that CRISPR functions as a bacterial immune system based on computational analysis. Then, in March 2007, Philip Horvath, Denisco, France, experimentally demonstrated that CRISPR integrates new phage DNA to fight off future phage attacks, confirming it as an adaptive immune system. Henceforth, as scientists continue to study and seek answers, finally, in January 2013, Feng Zhang successfully adopted CRISPR-Cas9 for genome editing in eukaryotic cells, showing targeted genome cleavage and programmable targeting of multiple loci. George Church Lab at Harvard University reported similar findings.
CRISPR-Cas9 technology has revolutionized Parkinson's disease research by offering powerful tools to manipulate genes implicated in the disease's pathogenesis. One significant application involves induced pluripotent stem cells, reprogrammed cells derived from a patient's tissues, such as skin cells. Researchers can introduce specific genetic mutations associated with Parkinson's disease, such as mutations in the LRRK2 or PINK1 genes, into these iPSCs using CRISPR-Cas9. This approach allows scientists to create cellular models that accurately mimic the genetic profile of Parkinson's disease patients. The study by Yoon et al. Represents a significant advancement in the application of CRISPR-Cas9 technology for treating Parkinson's disease. Yoon et al. Demonstrated the efficacy of CRISPR-mediated gene editing in targeting the A53T mutation in the SNCA gene, which is associated with familiar forms of Parkinson's disease characterized by early onset and severe progression. In their research, Yoon and colleagues used CRISPR-Cas9 to specifically delete the A53T SNCA mutation in a mouse model of Parkinson's disease. This approach led to notable improvements in disease pathology, including reduced alpha-synuclein aggregation, preserved dopaminergic neurons, and improved motor function. Notably, the study showed the selective targeting of the mutated SNCA allele while maintaining the function of the wild-type SNCA gene, which is crucial for normal synaptic function.
Now that we have an overview where we can alleviate the symptoms of Parkinson's disease through gene therapy, this vision is becoming a reality with tools like CRISPR-Cas9 and microRNA. However, as highlighted by Johan Kahn et al. In 2023, significant ethical issues arise, particularly in obtaining informed consent from patients with cognitive impairments. In medical practice, especially for Parkinson's disease, autonomy is vital. Yet, securing informed consent is challenging when cognitive abilities are compromised. Patients must fully understand the risks, benefits, and implications of gene therapy, including the handling of their genetic data. Beneficence strives us to provide effective treatments that enhance the quality of life for Parkinson's patients. When conventional treatments fail, gene therapy may offer hope, but it must follow strict guidelines to ensure patient safety, confidentiality, and dignity. Nonetheless, since the principle of do no harm requires us to balance therapy benefits and risks carefully, healthcare providers must offer clear information and robust support to families, maintaining patient privacy and preventing harm. Finally, the principle of justice demands equal access to effective therapies for all Parkinson's patients, regardless of financial or social status. It's about promoting fairness, impartiality, and equity in healthcare.
CRISPR-Cas9 is a gene editing technology that allows precise alterations to DNA within living cells. It consists of two main components, the Cas9 enzyme and a guide RNA. The gRNA or the guide RNA is designed to match a specific DNA sequence in the target genome, directing the Cas9 enzyme to that exact location. Once there, Cas9 creates a double-strand break in the DNA. The cell then attempts to repair this break using either non-homologous end joining, which can introduce insertions or deletions and disrupt the gene, or homology-directed repair, which can introduce specific genetic changes if a repair template is provided. This ability to target and modify specific genes makes CRISPR-Cas9 a powerful tool for genetic research and potential therapies, as demonstrated in Parkinson's disease research, where it has been used to create disease models and investigate gene functions. CRISPR can be delivered to target cells through several methods, each with unique benefits. Viral vectors like adeno-associated viruses, adenoviral vectors, and lentiviral vectors are popular for their ability to penetrate cells and provide stable, continuous expression of CRISPR components without integrating into the host genome. mRNA delivery offers temporary function and faster results, providing genome integration risk, as mRNA can be quickly transcribed. Plasmid delivery involves using plasmids for gene synthesis and transfer, allowing continuous expression without integration into the host genome. Lastly, protein-based delivery involves delivering the Cas9 protein as an RNA protein complex, which enhances specificity, reduces off-target effects, and increases efficiency.
Despite this, CRISPR still encounters a number of challenges and limitations, especially when it comes to clinical applications. One major issue is the risk of off-target effects or unintended genetic alterations at unintended sites. Researchers have made significant progress in enhancing CRISPR's precision by developing more accurate Cas9 variants and optimizing guide RNA designs. However, the necessity of inducing DNA double-strand breaks presents significant safety concerns, as CRISPR gene editing can lead to cell death, large base deletions, and chromosomal disorganization, potentially causing malignant tumors. Another limitation of CRISPR is the efficient and safe delivery of the editing machinery to target cells and tissues. For many genetic disorders, components such as Cas9 and sgRNAs need to be delivered to specific organs or tissues within the body, which is particularly challenging for internal organs or crossing the blood-brain barrier. Additionally, introducing CRISPR components into the body can trigger an immune response, leading to the destruction of edited cells or rendering the therapy ineffective. The risk was emphasized by the death of an 18-year-old subject in 1999 due to an inflammatory response induced by an adenovirus vector.
According to Sentence Summer in 2022, technologies like CRISPR-Cas9 and induced pluripotent stem cells or iPSCs are rapidly advancing our understanding and treatment of these conditions. iPSCs are revolutionizing lab models effectively mimicking diseases and using isogenic controls to minimize genetic differences between samples. This leads to more reliable and accurate research, helping scientists better understand diseases and treatments. However, iPSCs face challenges like genetic instability and tumorigenic potential. Similarly, CRISPR-Cas9, while powerful, can cause unintended DNA changes and delivery issues. To address these, researchers are exploring new methods like light-sensitive guides. Despite these challenges, clinical trials using iPSCs for Parkinson's disease and spinal cord injuries are underway, showing promise. While genome editing therapies are still developing, scientists are working hard to solve technical problems and establish consistent procedures.
Since the discovery that DNA changes cause cancer, scientists have sought simple methods to correct these changes through DNA manipulation. CRISPR has revolutionized cancer research and treatment by allowing precise and easy alterations to human cell DNA. Since then, CRISPR has shown remarkable potential in various medical applications. It has revolutionized research across diseases such as Alzheimer's, Parkinson's, Huntington's, and amyotrophic lateral sclerosis. The future of CRISPR in medicine is truly promising. Despite current challenges such as off-target effects and efficient delivery methods, ongoing research and development are paving the way for these advancements to become a reality. That is it for our presentation. Thank you so much.